Let us see how the synthesis of lac mRNA is regulated. So, two French investigators, Jacques Monod and Francois Jacob, performed a series of genetic and biochemical experiments in the late 1950s that elucidated the mechanism of regulation of lac system. They began by isolating constitutive E. coli mutants that make lac mRNA, means they make all the three enzymes, galactosidase, permease and transacetylase, in this presence as well as absence of the inducer because they are constitutive. Then they constructed a variety of partial diploid cells containing constitutive mutants and observed the cell's ability to synthesize beta-galactosidase. These mutations appeared to be of two types, termed as LAC-I and LAC-OC. The LAC-I negative mutations behaved like typical minus mutations in most genes and are recessive. Because lac mRNA synthesis is off in a lac-I positive cell and is on in a lac-I negative mutant, the lac-I gene is apparently a regulatory gene. The lac-I gene codes for a product that acts as an inhibitor to keep the lac structural genes turned off. The lac-I negative mutant lacks the inhibitor and thus is constitutive. A lac-I positive and lac-I negative partial diploid has one good copy of lac-I gene product, so the system is inhibited. Monod and Jacob called the lac-I negative gene product as the lac repressor. The most likely explanation for the constitutive lactose system is that the mutant strain synthesized a truncated repressor protein that cannot block transcription of the lac genes. तो ये जो mutant strains हैं, इनमें जो repressor बनता है that is truncated, जो कि आगे जाके lac genes की transcription को block नहीं कर सकता. This conclusion was confirmed when the lac repressor was purified and characterized. Genetic mapping experiments placed the lac-I gene adjacent to lac-Z gene and established the order of the genes as lac-I, lac-Z, lac-Y and lac-A. This was the order of genes. A striking property of lac-OC mutations is that in certain cases they are dominant. The significance of the dominance of the lac-OC mutations becomes clear from the properties of the partial diploids. Both the diploids lac-OC, lac-Z negative / lac-O positive, lac-Z positive and lac-OC, lac-Z positive / lac-Z positive, lac-Z negative are lac positive because there is a functional lac gene, lac-Z gene. In case of former, beta-galactosidase synthesis is inducible even though the lac-OC mutation is present. In case of former, beta-galactosidase synthesis is inducible even though the lac-OC mutation is present. An immunological test capable of detecting a mutant beta-galactosidase showed that the mutant enzyme is synthesized constitutively in a lac-OC, lac Z negative / lac O positive, lac Z positive, partial diploid, whereas the wild type enzyme is synthesized only if an inducer is added. This experiment takes advantage of the fact that a purified antibody to beta-galactosidase will also react with mutant protein as long as the structural differences between wild and mutant proteins are not too great. A reaction of this type, in which an antibody that is raised in response to one protein is used to detect a closely related protein, is called a cross reaction, and closely related protein is called cross-reacting material or CRM. Thus, the presence of CRM, which can be detected by a variety of standard immunological procedures, is indicative of the presence of mutant protein. Genetic mapping experiments showed that all the LAC-OC mutations are located between gene LAC-I and LAC-Z. So the gene order of the 5 elements of the LAC system is LAC-I, LAC-O, LAC-Z, LAC-Y, LAC-A. या आप कह सकते हैं IOZYA. ये जो है ये सीक्वेंस है लैक सिस्टम का. Together, these experiments lead to the conclusion that LAC-OC mutations define a site or a non-coding region of the DNA rather than a gene, and that the LAC-O region determines whether synthesis of the product of adjacent LAC-Z gene is inducible or constitutive. The LAC-O site is called the operator.